Hi everyone. My name is Hannah Lucy. So I am now a senior on the biomedical track. I'm going the research design. Like the research track, so it's a little bit different than most other co-ops that I have heard about. So I have done 3 co-op rotations. I'm a transfer student, so I don't need the 5. Anyway, so my first 2 were at Children's Hospital, and my first one, and it was in the Center for Autoimmune Genomics and Etiology. So we focused on research within autoimmune diseases like lupus. I was my main focus, and then also rheumatoid arthritis and like osteoarthritis, and different factors that contribute to those diseases. Mostly within your genome, like you'll have certain SNPs, so it's a single nucleotide within your genome that is different than a normal person's genome, and that could possibly be a large contributing factor to why you have that disease. So I'm sure you've heard about how you can like do genome editing, CRISPR, that kind of stuff. So I was like different delivery methods or into the body. So I was actually working on that. So I was responsible for optimizing different protocols for different, for like subcloning, site-directed mutagenesis, because I was actually creating my own Cas9 system, so a different genome editor, because we were focused on a single SNP and we did not have the correct Cas9 system in order to do that. So I was working a lot with that, and while doing that, I was also learning how to do different infections, transductions, like using lentivirals, or different things. It was really interesting. I had a really good time doing that. Learned so many wet lab techniques like PCR, just a lot of stuff. It was really good. And then also that same semester, I was responsible for trying to come up with a method for single cell cloning. So usually cells viability isn't very good in cell culture unless they're with a large group of cells. But human cells that aren't uh, infected with EBV possibly, which is the Epstein-Barr virus, uh, only very few survive. And so there needs, in order to really progress in this area, a method needs to be created to subclone single cells. That way, if that's all you have, you don't lose all your hard work. So that was another thing I was working with. So a lot of sterile techniques and just different troubleshooting protocols, lots of research. So yeah. And then my second semester at Children's, it was a little bit different. I did a lot more flow cytometry, and PBMC, like working with blood. So we would take whole blood from patients, and we would filter it out and isolate the different cells. And we were interested in B and T cells, because those are the main cells factoring into lupus patients, and we wanted to detect the presence or absence of the Epstein-Barr virus, because it's also a contributing factor. So we went through a bunch of different staining protocols, figuring out which antibodies and such work best for it in order to detect it. Lots of different flow, many different buffers, everything like that, going through the whole protocol. So that was really interesting. It was really neat. Learned so much. Worked with great people at Children's, and it was just an overall amazing experience. And then my last co-op that I just did this past summer was actually with a UC professor while I'm working in a biomedical acoustics laboratory. So we were focused on, well, I was focused on one major project that we recently just got a grant for, which was developing a simplified biofeedback system, ultrasound biofeedback system for speech remediation. So there's some children who have RSSB, which is a residual speech sound error. So like after they're 8 years old, if they've gone through a clinical intervention for 6 months, sometimes their speech disorder stays with them and doesn't get better, and it sometimes progresses. And so that's where these different kind of methods are starting to come in, with more of a biofeedback, visual, fun technique, because speech sound disorders are usually associated with other behavior or cognitive disorders, which makes normal conventional therapy not as responsive and more difficult for them. So I was working with that, and right now we're in the beginning stages. So we're just collecting data from all the different children, doing a lot of coding in MATLAB. So that's our main program that we're working with. And so we're trying to track the tongue when it shows up on the ultrasound screen. So it wasn't a GUI. We're like doing a lot of different codes and trying to track it the best using pixel intensities, Taylor series like curve, different just prediction like theorems and stuff like that for it, because the tracking quality is definitely the most important thing, and it needs to be automated. So that's where I was stepping in, was the automation part of it. So usually you would have to draw a region of interest or something around the tongue, and we don't want the clinicians to have to do anything. So automation was my thing. And then also towards the end, I was doing a lot of data analysis, looking for differences between the two groups of children, because you need to have a method to classify and just take say values that you get from the video, like from the GUI. So they're called normalized displacement values, because as you track your tongue, we do one production at a time of like a vowel consonant combination, like R, A, R. And we'll track the displacement values, so going from like midpoint of ah to the end of and while that happens, you see how much your tongue moves, like there's the different regions, we'll track that. And using those values, I'm trying to categorize whether it's a normal or disordered children by looking at the differences between them and finding different thresholds and optimizing and seeing if there's certain patterns like that are considered normal, because there's more than one way of producing R. So that's actually also my capstone, is doing that. So it's kind of nice just going straight from that into that, and it's part of a clinical trial, which is what I want to get into. So it was a perfect fit for me. Really lucky, and had a good time doing that. So thank you.